Hi, class, and welcome to the second podcast on Chapter 5. Remember, Chapter 5 is fairly big. It's got two parts. First part is going to be on the membranes, so the plasma membrane. We already went over that, and that's how material moves across that. And then the second part is going to be on enzymes and the laws of thermodynamics, in other words, how energy moves. Some big terms in this podcast that you want to kind of get a handle on would be that whole idea of thermodynamics, what a substrate is, optimum conformations of enzymes, and then another big one that you're going to want to understand is going to be free energy, or delta G, we sometimes refer to that as. And so let's get to work. Enzymes are going to be proteins, and so they're massive macromolecules, and they have a specific function. And so the one that we've used so far in a lab was called catalase, and what catalase did in our lab was it broke down hydrogen peroxide into water and oxygen. And so each enzyme is designed to break down a specific substrate. And so, for example, if you're lactose intolerant, that means that you don't have enough lactase, which is the enzyme to break it down. Sucrase is going to be the molecule that breaks down sucrose. Or remember when we talked about lipids and the fat substitute olestra, why is it a fat substitute and how does it work? We lack an enzyme to break down olestra, and so we're not able to get energy from it. So again, there's this idea that substrate is kind of like a key, and then the lock is going to be found in what's called the active site of the enzyme. And it has to fit perfectly in order for it to break down. One thing that'd be interesting to talk about is how does that actually occur? And so if this is the active site, the point at which we actually break down a substrate, it could be that it slightly, in this little micro area inside the enzyme, it could change, for example, the pH, or it could make a molecular tug on a substrate. Anyway, what it does is it lowers what's called the activation energy, the amount of energy required to start that chemical reaction. Okay, so let's talk briefly about the laws of thermodynamics. There are only two, so it's not that hard. And the first one's really easy. You learned it when you were growing up, and that is the idea that energy is neither created nor destroyed. And so when you drive a car or you put gas in your car, the energy from that gasoline used to be energy in the sun, and so it was absorbed by plants. Those plants were covered by dirt, eventually made these fossil fuels that we dig up. As you burn it in your car, that energy is going to be transferred to the engine, to the motion, to the sound, but it's not going to be transferred to energy. Now the second law of thermodynamics talks more about what happens to that energy. And as we move from one reaction to another, the second law of thermodynamics states that actually increases the entropy of the universe. Now, entropy is a big word, but what does it mean? It just means randomness. So each time we transfer that energy, we're increasing the entropy of the universe. What does that mean? All energy will eventually end up as heat. Now, how do you explain this? For example, this is the xylem and the phloem inside a plant. It's this beautiful structure. How could we ever make something that is so gorgeous and organized like that if at every step we're actually increasing the randomness of the universe? This is a little deep, but it's not that hard to explain. How are we able to get something so complex? Well, we do that. In other words, we get complexity in one area, but to do that, we actually have to even increase the entropy of the universe even more. Okay, so that's a lot of deep philosophical talk. Let's actually talk about how that applies. You learned a lot about energy, and maybe when you learned about potential energy and kinetic energy, or energy of motion. So a dam, for example, if you are a dam engineer, you are going to design a dam so that the water comes out at the bottom. And the reason you do that is it has more potential energy. If we let that water flow, this system right here is said to have a delta G of less than 0. What does that mean? It has the ability to do work. Now eventually it will level off, and now we have a delta G equal to 0. In other words, there's no free energy available. And actually, if we had to move the water from here to a higher level, then we'd have a delta G that's positive. In other words, we have to put energy in the system to get it out. Now, our body doesn't work like this. We don't release all of our energy at once. We actually do that step after step after step after step. So gradually releasing that energy. So all chemical reactions then we could put into two different types. They could be exergonic or endergonic. Now, you may have learned that as exothermic and endothermic, but a better way to talk about it is gonic, and gonic just means energy. So energy releasing or energy requiring. And so an example of a reaction that is exergonic could be, and the most important one in biology, is the breakdown of glucose into carbon dioxide and water in the presence of oxygen. And so we've got more energy within that glucose molecule than is contained in the products, and so we now have a system where delta G is less than 0. In other words, it's like having the water in the dam, and we're able to release that energy. Likewise, in the other direction, if we take carbon dioxide and water, to get that energy stored back in the bonds of glucose, we actually have to put energy into the system. And so photosynthesis is the exact opposite reaction. Now, the way it goes about doing this is not the exact opposite. They're way more different. Is that right? The way photosynthesis and respiration occurs is totally different events. But the reactants and the products and the products and the reactants are actually equal to each other. And as a result, respiration is going to have a delta G of less than 0, and that's going to be exactly equal to the delta G, which is positive in photosynthesis. In other words, where we have to go. So those are exergonic, endergonic. Now, normally, in order to get a reaction to go, in other words, let's say I want to burn sugar, and we were to do that in the laboratory, you simply do that by adding energy. And one good way to do that is to add heat. The problem with that is if you try to burn sugar inside our body by adding heat, it will melt. And so that's not great. And so that amount of energy required to get the reaction going is what's called activation energy, or we refer to that E sub A. Now, with an enzyme, we can actually lower the activation energy, and so we can have a lot of those reactions going on at a temperature that's not damaging to the tissue. And so the cycle occurs like this. If this is sucrose, remember this is a disaccharide, that comes into sucrase, and that allows it to break that up. Now, why do we have water coming here? This is hydrolysis. In other words, we're adding that water, and we're breaking that sugar down into monosaccharides. Now, if you look at the enzyme, it continues and continues and continues. In other words, it's used over and over and over again to break down those substrates. And so it's not modified, but it's making a site or an active site where it's able to have that occur. If we lack the enzyme, then we can't break it down. Now, each of these are going to have an optimum pH and optimum temperature. So, for example, in us, the optimum temperature, obviously for all the proteins inside our body, are going to be right around 37 degrees, or that's going to be right at body temperature. So they're designed to work optimally at that. Now, why don't they work as well when it's really, really cold? What we find is that it doesn't change the structure as much as it changes the amount of times that molecules are actually able to get into that active site. And so how do molecules get into the active site? They move about, and if they bounce into the active site, then they'll be broken down. And so as we increase the temperature, we increase the molecular motion, so we're going to have an enzyme that's going to be working more efficiently as we increase temperature of the whole reaction. But each of those is specifically modified to where it exists. And so if you look at it getting too hot, then eventually we denature the protein, so we change its shape, and then it can't function anymore. Now, let's say you're a bacteria that lives in Yellowstone Park, where it's near boiling. Then you want to have proteins that are optimized to that. And so no matter where you live, you're going to have proteins that have optimized through natural selection to that environment. Now, the same thing occurs for pH. So, for example, pepsin is an enzyme that breaks down proteins in our stomach, where it's really, really acidic, and trypsin does the same thing, but it does that in the small intestine, where we have a basic environment. And so each of those are modified specifically to where they are. Now, the last thing that you should kind of have an understanding of is how do we regulate that? In other words, how do we make sure that enzymes are breaking down materials when we want to and not breaking it down when we don't want to? If you think about it as how could I stop this from breaking down molecules, there's essentially two things that I could do. I could, number one, block that active site. And so we call that competitive inhibition. In other words, if we add something right here that blocks substrate from moving in, then we can block that reaction from occurring. That'd be one way. The other way is to somehow change the shape. And so we could add another chemical down here on the enzyme at the bottom, and that changes that active site and where it is, and so we can control that. If we then remove that, it goes back to the shape it was before, and so it's going to break it down. So really sweet little regulation in enzymes. It's very important in maintaining all those chemical reactions inside our body.